The TRANSCEND-001 study is a study of lysocaptogene maraleucel, otherwise known as JCAR017, for relapse refractory aggressive B-cell lymphomas. This is an anti-CD19 directed CAR T-cell product with a 4-1BB co-stimulatory domain. And unique from other CAR T-cell products in the space, the CAR T-cells themselves are not manufactured from bulk T-cells, but rather the CD4 and the CD8 T-cells are separately expanded and transduced and then delivered in a 1-to-1 ratio with to the patient. The rationale for this is largely based on animal model data performed at the Fred Hutchinson Cancer Center in Seattle. And when they looked at giving either a CD8 or a CD4 product versus a ratio of combined CD8 to CD4 CAR T-cells, they found better persistence, expansion, and anti-tumor activity. And when they looked at different ratios, they found that the 1-to-1 ratio of CD8s to CD4s appeared optimal. It is on that foundation that the initial phase 1 study and then the multi-center TRANSCEND study was designed. So that study is ongoing. It's a seamless design that's leading to a pivotal cohort which is currently expanding. What we reported on today are the first 2 segments of that study, which was a dose finding and escalation study, and then a dose expansion study. In the dose finding and escalation study, we've treated patients at 3 different dose levels and schedules. We have a first dose level, which is 50 million cells administered a single dose. We have a cohort of patients who we treated with the same dose of 50 million cells at 2 doses given 14 days apart. And we have a second dose level of 100 million cells administered as a single dose. Based on what we saw in that dose-finding cohort, we expanded 2 of those dose levels, dose level 1 and dose level 2, both given as a single infusion. We saw no apparent benefit to giving a second dose, so we're moving forward with just single infusions. Based on those data, we've now initiated accrual to our pivotal population, which is ongoing right now. So today, we reported on 91 evaluable subjects in the dose-finding and dose expansion cohorts. These patients included diffuse large B-cell lymphoma that were relapsed or refractory to at least 2 prior lines of therapy, included DLBCL NOS, DLBCL that was transformed from follicular lymphoma, CLL or marginal zone lymphoma, as well as primary mediastinal lymphoma, grade 3B follicular lymphoma. We also included double and triple hit lymphomas, which are classified as high-grade B-cell lymphoma. Among those patients, the majority were DLBCL NOS, so 3/4 of patients had DLBCL NOS and about 20% had transformed follicular lymphoma, with the small remaining being a balance of the other less common histologies. The safety profile was excellent in our overall population. The most common toxicities were cytopenias. And this is common in a product where you give fludarabine and cyclophosphamide lymphodepletion prior to giving the CAR T-cell product. And so we saw high rates of neutropenia, anemia, and thrombocytopenia, but those do recover, though it can take some time. Other toxicities were actually uncommon, and we saw very little in the order of grade 3-4 toxicity other than cytopenias. Adverse events of special interest around CAR T-cells include cytokine release syndrome and neurologic toxicities. We saw an overall very low rate of cytokine release syndrome. Only 35% of our patients developed any cytokine release syndrome, and with 91 evaluable patients, we only had a single case of grade 3-4 CRS. So virtually all of our cytokine release syndrome was grade 1-2 and was entirely reversible. Our incidence of neurologic toxicity was also relatively low at 19%, with a 12% incidence of severe neurologic toxicity. Once again, all severe neurotoxicity was entirely reversible. There's only 1 case of ongoing neurotox, and it's a grade 1 tremor that is ongoing at the date of the last follow-up. So overall, I'd say this was extremely well-tolerated therapy. We saw no deaths related to CRS or neurotoxicity, and we saw no cerebral edema. Efficacy, of course, is what people really want to know about, and this was highly efficacious therapy. So if we look in our overall population of patients, we saw a response rate of 74% and a complete response rate of about 54% in the entire population of patients. If we then look even 6 months later to assess for durability, 1/3 of our patients are in ongoing remission at that time point. We looked at different histologic subsets and found that the DLBCL NOS patients, transformed follicular patients, responded very well to their therapy. We saw lower rates of response and durable response in patients with Richter's transformation or transformed marginal zone lymphoma, though those were a less common subset of patients. Only 10 subjects overall met those criteria. We're now accruing our pivotal cohort of patients, and that population is limited to DLBCL-NOS and transformed follicular lymphoma, as well as high-grade B-cell lymphoma with double or triple hit cytogenetics. Our pivotal population is also limited based on certain eligibility criteria. We only accrue 0 to 1 performance status patients in our pivotal study, while our prior study included patients with a 0 to 2 performance status. We allow prior allogeneic stem cell transplant in our entire dose finding and dose escalation population, but limit our pivotal study to ECOG patients with 0 to 1. If we look within our overall accrued population to those patients that meet our pivotal study definitions, we actually higher rates of overall and complete remission rate. And in that population treated at dose level 2, which is our pivotal dose going forward, half of patients were in ongoing complete remission beyond 6 months. And most patients who achieve a CR at 6 months have remained in CR beyond then. So we're very encouraged by the results so far. We're encouraged by the toxicity results that appears manageable and low in incidence. And obviously the response rates are very exciting with this product, as with other CAR T-cell products under investigation.